But, okay, so this is what I'm doing now, but what I wanted to tell you is a little bit about my PhD work, and this is mainly because when I was going through all these pictures that I collected for Monday evening, I accidentally bumped into my PhD lecture. So I thought, okay, this is probably the last opportunity I will have to present some of that work. And I stared at it. I didn't remember anything at the beginning. But after a while, it all came back to me. So, and what I've done along my career path, I would say, is that I almost completely changed my my interest, but I stayed. The only thing that I could think of is that I still really, really like membranes. So I'm still working on membranes and membrane deformations, but in a very different methodologies and different protein machine or complexes. So this is as is, okay, so that was the title of my PhD talk, and it was about the interplay between the alpha and beta gamma subunits in the signal transduction cascade in the Drosophila visual system. So we heard during that week several talks about the achievements of Tzvi in the GTPR cycle and biochemistry of G proteins. And then the second half of the science where he worked on the Drosophila signal transduction cascade. And I came pretty late to the lab, and I merged together with Mickey, who is organizing the meeting. Actually, Mickey recruited me to the lab. And then I joined a project, and let me just show you pretty much what we did. So I want to start by saying that the experiments were very primitive. We took flies, we put them on the fluorescent light on the bench. That was how the experiment went. And then we put them for some time in the dark and sometimes in the light. It was a very, very simple experiment. Like, this drawing is not simplification of the assay. This is how it was. And then we pretty much squeezed, like smashed the flies' heads to make a homogeneous. That would be the scientific way to describe it. And then we separated the pellet fraction from the supernatant fraction. And we looked to see the localization. And specifically, we started by looking at the localization of the G alpha subunit. That's GQ alpha. And as you can see in the flies that were adapted to dark, most of the protein, or almost all the protein, was in the pellet fraction, so on the membrane where it should be in order to transduce the signal. But after illumination, we saw like a gradual translocation of the alpha subunit from the pellet fraction to the cytosol. And these results were almost fully there when I joined the lab from experiments that Mickey did. And then the explanation that we had for that is that we knew that the alpha subunit is lipidated by palmitoylation, and that there is a meristylation moiety on the beta gamma subunits. And the idea was that upon activation, we have depalmitylation. So there will be a thioesterase, palmitoyl thioesterase here, which will cleave that bond. And then we have depalmitylation that will lead to translocation of the alpha subunit from the membrane to the cytosol. Okay, then we asked, so that was all nice. We put the flies back into the dark. The alpha subunit went back to the membrane where it can interact with rhodopsin. And then we asked the question, okay, so what is the role of the beta gamma complex in that signal transduction cascade? And we had several mutants of the beta subunit in Drosophila. I should say that all this work was in full collaboration with Baruch Minke. And then we had... and these are... Charles generated the mutant. I told you that I don't remember much about these days. And so we had the kind of a heterozygote mutant of kind of 50%, I guess, expression of the beta subunit. And then we have two hypomorph mutants, the beta E2, which was which has small levels of the beta subunit, and then the beta 1 was the most reduced levels of the alpha subunit, of the beta subunit. And when you look at the localization of the alpha subunit, you could clearly see that in the absence of the beta subunit, most of the, or almost all the alpha subunit is in the cytosol. So clearly, you need the beta gamma complex in order to be associated with the alpha subunit in order for it to be membrane bound. Okay. But then we did the electrophysiology of that of these mutants, and surprisingly, maybe not surprisingly, what we saw that these are all recordings done in dark state. So these flies were not subjected to light, and while there is almost no spontaneous activity in wild-type flies, in the heterozygote mutant we had massive spontaneous activity. So again, these are in the dark. If you reduce the beta levels even further, you get less and less of this spontaneous activity. And I just want to remind you, a slide back, I showed you that there is less alpha subunit on the membrane, so there is less protein available for activation. So how come we get more activity, more spontaneous activity when we deplete the beta subunit? And now I'm just cutting a lot of data and experiments, but the point was, which I think was, for me, it was a really interesting finding and something to think about, that the whole point here was stoichiometry. So what we ended up finding is that you need a twofold axis of the beta gamma subunit over the alpha subunit, and we think that the physiological role of that was to suppress any spontaneous GDP-GTP exchange mediated by the alpha subunit. So if you have access, every alpha subunit that translocated to the cytosol and underwent this GTP-AS reaction and is bound to GDP, if it is not immediately bound to the beta gamma subunit, you will there will be some spontaneous exchange to GTP, and then you'll end up with spontaneous activation. But if you have a stoichiometric axis, then you can ensure that the minute the alpha subunit is charged with GDP, it will bound the beta gamma subunit, which will stabilize this conformation, and then it will prevent spontaneous activation. And we indeed what we did in order to confirm that is that we reduced the level of the alpha subunit in the beta heterozygote mutant by 50%, and as expected, we completely abolished the spontaneous activity. So we restored the 2-to-1 ratio, and a spontaneous activity was back to normal. So this is something that I must say that I'm really proud of for my PhD work. I just want to end by acknowledging the people. So this was a really exciting, very exciting time for me working with Tzvi. I've worked very closely at the beginning with Miki, who really introduced me to lab, to experiments, laboratory work, you name it. I did my PhD together with Yael Litvak, who's now applying for positions in Israel. So if you are interviewing her, at least I can say, not because of me, but because of Tzvi, that she got really good education in science. Collaborators, so it was a really close collaboration with Baruch, which was very exciting because it allowed us to combine, and this is also what I try to do today in my lab, to combine biochemistry with the Drosophila genetics and with electrophysiology and EM, which we ended up doing. So I was able to combine all these techniques into some biological understanding of signaling. And just a little bit, say a small thing about Tzvi, and as we called it on Monday, the family of Tzvi, because we remain in very good touch after, when I came back to Israel and applied for position, Tzvi already passed away, and it is really, I just want to take this stage and thank Baruch and Dani who really helped me a lot and were kind of and adopted me in all these very stressful process. Okay, so now back to science. So that was also science, but back to what I do these days. So then, of course, I decided to take an adventure and change my field completely. And I went to the lab of Jennifer Lippincott-Schwartz when I wanted to study things on the single cell level, all these bands, you know, when you average all that, like tons of cells in one band, it kind of bothered me at some point. Now I miss it a little bit, I must say. But then I joined Jennifer's lab, and at the beginning I actually worked on microtubule acetylation. I really still liked post-translational modifications, and I heard that microtubules undergo acetylation, and the role of tubulin acetylation was unclear. I said, okay, let's do that. And one of the major phenotypes that I had is in this last stage of cell division. So in cell division, if you just consider the membrane for a minute, then you have this acute actomyosin contraction that pretty much separate the cytosol into two huge domains. But then the cells end up being connected to one another by this 1 micron diameter bridge. And then this membrane has to be cleaved eventually in order to give rise to two fully separated cells. So the mechanism for that was unknown, and I had mutant of acetylation where that was delayed, but I was unable to find the connection between acetylation and the delay in abscission, and I was kind of in a phase in the lab that I didn't know where to take that project. And then one day, Jim Hurley came to the lab and said, I just crystallized a complex of CEP55, which was a protein that localized to this bridge with one of the ESCRT components that is Alex. And he said, you know, based on the crystal structure, I made some mutations. And he went to Jennifer and said, can someone help me with that? I just want to test it in cells and see if there is any phenotype. And I said, okay, I'm looking at these bridges anyway, and I don't know what to do with my life. So I'll just take these mutants, and what the hell. So we ended up publishing that in Science. That was a really nice coincidence. But then what I really realized is that first, we don't know the mechanism for this membrane cleavage event. And second, these ESCRT proteins are super cool. So I decided that I will follow up on that, and that is what I actually do until today. So because this is a signal transduction meeting, maybe I should say, so the ESCRT machine is actually a machine that mediates membrane bending and fission, and the mechanism for that is unknown. It is involved, now we know that it is involved in many, many processes, but the classical processes is first the multivesicular bodies biogenesis. So in one of the talks, it was mentioned that the receptor can continue to signal after it was internalized into endosomes. So one of the mechanisms to prevent that is internalization of the receptor into this endosomal membrane, that giving rise to this multivesicular bodies formation, and this is mediated by the ESCRT complex. The HIV and other retroviruses hijack the ESCRT complex in order to bud out of cells. And we, and together with other groups, have shown, and I'll show you some of the data, that they mediate this last event in cytokinesis, that is abscission of this membrane and microtubule based bridge. So I pretty much said that. So just to show you, this is some slide from still from my postdoc work. What we've done initially was just to characterize that in a very high spatiotemporal resolution. We wanted to see how the process really looks like. And the first thing that we saw is that this is kind of a 20-minute event. And what we see, these are the microtubules. You can see gradual constriction of the microtubule and until you end with this fission event. If you look at one of the ESCRT components, you can see the movie will loop in a second. So you can see that escorts are not there, and then they jump on to the bridge exactly where you see, at the same time that you see constriction and fission. So that was the first indication to us. If they're there in the right place at the right time, they probably are the ones that convey the job. And just to give you a brief introduction on ESCRT, so as this is a family of proteins, these are all cytosolic proteins that are recruited to the membrane upon a signal. So we looked at the different components of the system, and we analyzed the kinetics of their arrival. And what was clear to us is that this ESCRT complex is the one that rapidly elevates right before abscission, and then we have the cutting of the microtubule. So we knew that there is something really interesting that involves these ESCRT components. And I should tell you that now we know, we didn't know it back then, but now we know that this ESCRT component is evolutionary conserved all the way from archaea, and it is actually the complex that mediates cell division in archaea. So, but back then, I think that the papers were published pretty much at the same time or very close to it. Okay, so then I was in Jennifer's lab, and the next thing I wanted to know, okay, so we have this temporal recruitment of different ESCRT components, but where exactly do they localize to? And that was important to me in order to understand how this constriction event is actually mediated. So what we saw, this all was done with structured illumination microscopy. There were no commercial structured illumination microscopes at the time, so I've done everything on some kind of a beta version somewhere in upstate New York. But what we've seen was really interesting. We saw that the first ESCRT component, called SEP55 in abscission, formed like this disc shape. Then if you look at ESCRT1, we found two really adjacent rings located at the center of the bridge. And then ESCRT3 had similar rings, but also had this a small localization, and actually we ignored that for a very long time. This small localization exactly at the constriction site. Okay, so at the end of my my postdoc work, we had the temporal recruitment, and we had some spatial information. We knew that there is an array of rings, and that at some point late in the process, we have recruitment of this ESCRT3 component and this is like a half a micron away. Okay, so this is kind of in terms of proteins, this is pretty far away. So then I started my lab thinking, how do you get from this stage to this stage? Okay, and this is, and I want to pause for a second and say that this is pretty much what we do in the lab. In the lab, what we are trying to do now in the lab is to understand the mechanism, the fundamental mechanism of membrane fission by using abscission as a model system, and by applying high-resolution imaging techniques. But at the same time, we are trying to push the limits of the microscopy techniques. And I think that, at least so far, this has been really beneficial to us, because we have a problem, and this is why we have the need to push the technology further. And then this is kind of a circle that I find very, very useful. And my lab is kind of split. Half of the lab is actually working more on technology development, and nowadays we are mainly focusing on labeling. And half of the lab is asking more biology, cell biology questions. But I think because this group of people is always interacting with one another, we often get to some crazy experiments and fun things. So this is where we were. And then, as I said, at this point, we knew that the ESCRT3 complex and the VPS4 ATPase, which I didn't mention until now, are the evolutionary conserved minimal functional unit for mediating membrane fission in general. But this story is not a simple story, because this is not a homopolymer, the ESCRT3 complex, but rather a heteropolymer We have over 10 different ESCRT subunits, and we do not know the stoichiometry of the subunits and also the composition of the filament in different physiological contexts. So this is still a mystery to us. Okay, so from in vitro data, we knew, and from data also in cells, we knew that if you take components of the ESCRT complex, you see these spiral-like, this is negative staining EM, spiral-like structure, and if you add lipids to them, they are really sticky to lipids. So they can bind membranes, and they form these helical filaments. If you overexpress them and you unroof the membrane and you look on the inner side of the plasma membrane, you also get these spirals. But this is not under physiological condition, but rather under really high overexpression levels. In abscission, we and others could see these filaments going all the way from the center of the structure to where abscission occurs. But we didn't know whether these filaments are ESCRT filaments. So for that, we decided to go with a 3D storm. So the advantage of storm is that we can actually label a specific protein. Now we can look at a specific protein, and then if we see some interesting structures, we know for sure that this is the protein. And I can tell you that our group and other groups, the group of Daniel Gerlich, we put a lot of effort into labeling ESCRT components into these in these EM tomograms, and we weren't able to do it. Okay, so Ina, a very talented PhD student in my lab, decided that she will take on this project. And this was very challenging at the beginning, and I'm not going to show you that data, of course, because in order to get structural information from storm data, you need very high density labeling, and this is really hard to get many times. So she screened for tons of antibodies until we got lucky, or not lucky, with one of the antibodies, which is the IST1 antibodies, which is one of the ESCRT components. And what we started to get is these helical filament structures. So this is one structure. This is the bridge, okay, and this is the filament, and the color here is basically height. Okay, so that indicates the height of the localization point in the structure. I'm waiting to do this. Okay, and then, no, the other way around. Okay, and then we also saw, oh, thank you. Okay, this was really, I remember, I must tell you that Ina sent me these images, and I was running through the corridor showing them to anyone who was there in that hour. But this is really the first spiral. Now we have several of them that we saw that stretched all the way from the dark zone. I didn't mention that word before, but from the center of the structure where these initial rings were shown before, all the way to the constriction or to the abscission site. So at this point, we already knew that we have a helical filament that is stretching all the way from the center to the constriction site. And then there was another phase where we just collected lots of data. And what we wanted to do with that is first to draw the sequence of events. So how do you get from this initial ring to these two pools that I showed you by structured illumination? And we've done that. We put a time sequence on these in two ways. First, we compare that to live cell imaging. And second, we took all the data, and because we knew we are dealing with a constriction event, we just measured the ratio between the diameter here and the diameter here. And that gave us an indication for how progressed we are in this constriction process. And you can see that data, this is just divided by the stage. So you can see that here the bridge is not constricted, and here the bridge is fairly constricted, indicating that this is the late stage. And we had a very interesting stage. This is stage 3, which with more data, I hope that we will open it to more stages. But we knew that something really interesting happens here, and that this stage is involved with some initial constriction. So I won't bore you with all the details. I will just say that based on that, because we can actually follow this structure, we can actually calculate the polymer length. So what is one of the questions that we were dealing with is, do we have polymerization from this transition from stage 3 to stage 4, or do we just have stretching of the filament? So we calculated that, and that was done in collaboration with Misha Kozlov from Tel Aviv University. And what we have clearly seen is that the transition between stage 3 and stage 4 involves polymerization, but then we have a depolymerization phase that actually, which is actually the stage that gives us this acute constriction. And this is our current revised working model. So we think that the process starts with assembly of an initial ring of the ESCRT complex that is formed on the backbone of ESCRT and ESCRT, and then we have a polymerization phase. Then we have stretching of the filament that involves some small constriction. But then at some point, and this is what we are working very hard to understand nowadays, we have acute constriction of the filament and distortion. You see that this is probably under a lot of tension, and we think that this is because it is still bound to the scaffold here at the center, but it is free to relax and constrict on the edge. And I will just say that we think that this involves microtubule severing. And then eventually, because this form is under a lot of tension, it breaks or depolymerizes, and then we end up with two pools, one that is stacked to the scaffold of the other escorts, and one that is constricting all the way to 200 nanometer. So this is 200 nanometer. This is not full fission. What we are trying to do now is to develop technologies to open and to to really resolve the 200 nanometer to zero. We started using expansion microscopy. We'll see. We want to combine that with super resolution microscopy, and then we'll see where it will take us. So then the fun thing about it is that now we can actually apply a structure function approach. We can take different mutants of different escort proteins and say, how do they interfere with this scenario? And I'll just give you a few examples. Of course, we've done the kinetics of all of them. They all have very severe phenotypes in abscission. One nice thing that we've seen is that if you deplete the most, this is CHIMP4B is the most fundamental evolutionary concept and abandoned ESCRT component. What we found is that if we deplete it even to very low levels, we still get some filamentous structure. Of course, they have a very deformed morphology, but it appears that IST1, which is the protein that we are looking at, has some intrinsic polymerization features, even in vivo. It's been shown in vitro, but everything we do is inside cells. Another example is that we depleted the VPS40, the AAA ATPase, and what we see is that we do get filaments, but these filaments mislocalize. So instead of being localized from here to here, now they just localize in the center, and they are unable to induce constriction. Another interesting example is CHIMP2B, another component. So now we can form the filament, but the filament is not bound to the membrane, and it is, again, unable to induce constriction. It looks like it lost completely its rigidity, and I should tell that from work done in artificial lipid, it's been shown that the CHIMP2B really changed the rigidity of the membrane lipids. So I want to end this part, but just by showing you a cartoon of how we think, this is something we recently made for the lab, how we think this process is occurring. So we think we have this initial escort organizing, and then on the backbone on escort 1 and 2, we have polymerization of the ESCRT complex, which is composed of different subunits, and then we have deformation of the helix, microtubule severing, and constriction. So there is a lot of speculations here, but I think we're getting to, I like to dream, so at least we can dream about how this thing works, and if we are wrong, we'll just draw new models. So for the second part of my talk, So this is the summary. I think we can skip that. So so this was all very structural, and we think we can retrieve some dynamic sequence from work on a fixed cell, but what we really want to do is do everything on live cells. But the current state is that super resolution techniques are very limited in their temporal resolution for various reasons. And on top of that, we know from ESCRT specifically, and it's been shown for other proteins, that conjugating a protein as large as GFP is associated with some artifacts in dynamics and in localization. And this is even more severe when you deal with polymers, right? You have a lot of protein-protein interactions that has to be taking place at the same time. And if you add these 27 kilodalton protein, that can interfere with that. So we want to do something more elegant. And the way we approach that is with the click chemistry and genetic code expansion. So the idea is that you take a protein and you modify one of the amino acids to what we use is a modified lysine, and then with the right chemistry, this is with a tetrazine, we get a covalent bond with a very minimal tag. So we have a di tag to a tetrazine, which is cell permeable. We add it to the cells, and then we can get a really direct, almost tag-free labeling. This work was pioneered by Peter Schultz and was then followed by Jason Chen. And the idea is that you mutate one of the amino acid codons into a THG, and then instead of it being encoding for a stop codon, you engineer the cells so that now you incorporate this modified amino acid into the protein sequence. And then, as I said, you have this bioorthogonal chemistry or click chemistry where you can bind the dye directly. So, of course, we need to engineer the cells. You have to introduce a tRNA and a tRNA synthetase and the amino acid. We do all that with in a very close collaboration with the lab of Elia Lab at Gurion University. And we started, this is just to show you kind of, for those of you who are not familiar with the technology, kind of a very quick, intuitive assay. So you take an M-cherry protein separated by a TAG stop codon and then a GFP. So now if you have translation of the entire polypeptide, you have red fluorescence and green fluorescence. And this is only when you add the unnatural amino acid. If you do not add the unnatural amino acid, what you end up is with just red fluorescence, because this encoded a stop codon. And then you stop translation in the middle of the protein. Okay, so this is the basic principle of the approach. And we started because we wanted when I wanted a benchmark protein where we can actually compare our measurements to. So we decided to take alpha tubulin. That ended up being a really bad choice, but we managed to do it eventually. And what we've done here are highlighted the sites, because the first thing you need to do is to find appropriate site that will not interfere with the protein function and cell biology. So we screened from four different sites, and this is just the Western blot. And you can see that this is with amino acid and this is without amino acid. So you can clearly see that all of them worked, some of them with better efficiency than others. Here, if you look at the 280 side, you can see that we have this truncation form. And this is because no matter what we do, the process is not 100% efficient. We always get some of the TAG functioning as a stop codon. Okay, so at this point, we knew that we can incorporate the modified amino acid. Then we started testing the click chemistry, and we have done that. Most of the work that we've done is with the silicon rhodamine tetrazine, which is a dye that was synthesized by Kai Johnson. And the first thing was to see that we do get labeling, and this is a lesson for people that are interested in using that the efficiency of incorporation of the modified amino acid doesn't tell you anything about the efficiency of dye binding. different dyes have different chemical properties, and they can bind with different efficiencies. So that was the first lesson that we learned. So some mutations that were very efficient were much less efficient in terms of specific labeling. But we managed to find good sites, and this is the movies of microtubules directly labeled with silicon rhodamine, essentially tag-free. Okay, this is just, we've done a lot of validation. It is all already published work, but this is silicon rhodamine labeling, and this is tubulin GFP, just for comparison, and we could see that